Okay, so on Friday we started talking about the SN2 reaction. Today we'll continue our discussion about various characteristics of the SN2 reaction, and then we'll start talking about the SN1 reaction. So at the end of class on Friday, we started talking about stereochemistry in SN2 reactions. And I showed you the results of an experiment which demonstrated that the stereochemistry is inverted at a stereocenter that undergoes an SN2 reaction. In order for that inversion to occur, the nucleophile has to approach that carbon from the side opposite of where the leaving group is. And so we refer to that as a backside attack. Okay? And this is a characteristic, a general characteristic of SN2 reactions. They proceed via inversion of configuration due to the backside attack. And we see this here in this very nice figure from the book. So here on the left, we have our chiral alkyl halide. The one they've chosen here has a deuterium and a hydrogen. Those are different isotopes, so they're different groups. So indeed, this carbon does have 4 different groups attached. And as the nucleophile approaches 180 degrees, this is supposed to be a degree symbol. Somehow this got messed up in this figure and I couldn't edit it. As the nucleophile approaches 180 degrees opposite from where the bromine is, then the 3 groups that are pointing towards the nucleophile are going to start to move away from the nucleophile. We're going to start to form the new bond between the carbon and the nucleophile while simultaneously breaking the bond between the carbon and the bromine. And so when we get to the transition state, we have this trigonal bipyramidal arrangement where we have the 3 groups that are not participating in the substitution reaction coplanar with each other with trigonal planar geometry. And then we have the nucleophile and the leaving group 180 degrees opposite each other. And as we proceed to the products, the bond between the carbon and the nucleophile gets shorter and stronger. The bond between the carbon and the leaving group, the bromide, gets longer and eventually breaks. And then those 3 groups, as that process continues to occur, those 3 groups are going to move away from the nucleophile until they reach the tetrahedral geometry that we have at the product. Okay? So the inversion process involves the nucleophile approaching from the backside of the carbon relative to the leaving group, combined with the orientation, the position of those 3 groups flipping, kind of the same way that an umbrella will invert in a windstorm. The position of those 3 groups on the carbon will invert during the substitution process. Okay? So this is how the process occurs, and we know that it does occur because of the experiment I showed you on Friday, whereby measuring the optical rotation of the product that was obtained, it was clear that the stereochemistry had inverted. But what we need to understand is why. Is that what you were asking, or are you going to ask something else? Go ahead. Right. So now we're going to explore why that is the case. Why is the nucleophile coming from the backside? Why is it not coming from the same side? If you look at it, coming from the backside, you have these 3 groups pointing at the nucleophile. So you would think there's going to be more steric hindrance if the nucleophile comes from the back, as opposed to coming from the front, coming on a trajectory close to where that leaving group is. But there must be a reason. So why? Why is it happening via backside attack? One way to think about it is, why wouldn't it happen by front side attack? What would be bad about having a front side attack? Yes? Okay, so the bromine is a big atom, it's true. What does the bromine have? A partial negative charge, right? The bromine is Lewis basic. The bromine has a partial negative charge. Our nucleophile is also a Lewis base. In most cases, it has a full-blown negative charge. Even when it doesn't, it has a lone pair. It is electron rich. So a big problem with a front side attack is that you would bring two electron rich groups in close proximity with each other, and they would repel each other. Okay, so it is that electrostatic repulsion of the nucleophile in the leaving group that prevents a front side attack and leaves you with the option of a backside attack. There's another explanation for why backside attack occurs, and it involves molecular orbitals. When I taught you about molecular orbitals in chapter 1, I said that we're mostly going to use molecular orbitals in 352M, but there are one or two cases where they're useful in 351M, and this is one of those. So when we were talking about anti-bonding molecular orbitals, I told you that if you introduce electrons to an anti-bonding molecular orbital, you will break the bond that is associated with that molecular orbital. So each of the sigma bonds in our starting material has a sigma star or an anti-bonding molecular orbital associated with it. And if you have electrons on a nucleophile overlapping with that empty sigma star orbital, you're going to initiate the bond breaking of that sigma bond. And it turns out that the location of these anti-bonding molecular orbitals in space is on the back side. So the anti-bonding molecular orbital of the carbon-bromine bond is located in the backside. And so as the nucleophile approaches the backside of the alkyl halide, the electrons from the nucleophile are going into that anti-bonding molecular orbital, initiating the bond breaking process, and ultimately getting us to the product. Question? That's a great question. It is due to the fact that it's an anti-bonding molecular orbital in the starting material. To go from the starting material to the product, we have a reorganization. We have a breaking of some bonds, making of other bonds, and so the orbitals undergo a reorganization process. And so that sigma star molecular orbital is gone in the products because the bond associated with it is gone. Okay? There's a new sigma bond, and then there's a sigma star associated with that new bond over there. Okay, so that was a great question, and the answer is that we're not forming a bond with a sigma star. What we're doing is initiating a bond breaking process, and then we have a reorganization of our molecular orbitals. Okay, other questions about the backside attack, the inversion of stereochemistry? All right, so now we have two ways to explain why the inversion occurs. We also need to be able to draw the products of an inversion. And the way we draw the products depends on how the starting materials are drawn. This sample problem from the book shows you how to show inversion of configuration if it's a cyclic alkyl halide. You see here the leaving group, the bromide, is cis to the methyl group. And our nucleophile, cyanide, is going to approach from the backside. And because it will invert the stereochemistry here, the cyanide is going to have to be trans to our methyl group in the product. And so we see that over here, methyl group, cyanide, trans to each other. So that's how we show the inversion when we have a cyclic alkyl halide. When we have an acyclic alkyl halide, those are usually drawn in one of two ways. This is less common, but we'll show it to you first. If we have the leaving group, the bond between the carbon and the leaving group drawn in the plane of the paper or the screen, then usually the easiest way to draw the inversion is to draw the mirror image of this, but not with the leaving group there, with the nucleophile replacing it, right? So this is not a mirror image of that because SH is not the same as I. Right, but the other groups are mirroring. The other three groups are mirroring each other, and we've just replaced the leaving group with the nucleophile. Okay, so that's usually the way we would do it if the leaving group is drawn in the plane of the paper. But it's more common to find the chain drawn in the plane of the paper in our typical staggered conformation. and then have the leaving group coming either out of the paper or the screen with a wedge, or going back into the paper or screen with the dashes. And there we simply draw the nucleophile with the opposite wedge or dash relative to what the leaving group was shown with. So here, the leaving group is coming out of the screen, pointing out at you. So the hydroxide nucleophile is going to have to come from the back side. So in the product, we draw the hydroxide with the wedges, or sorry, with the dashes. And then note that the position of the hydrogen atom that's just spectating in this reaction is going to switch from dashes here to a wedge here. Okay, so 3 different ways of drawing the product of an SN2 reaction relative to the starting material, depending on how it is drawn in the starting material. Questions? Yes? We will know that by the end of this chapter. So that's something that we will address late on Wednesday and early on Friday. How to distinguish between SN2 reactions and SN1 reactions based on the nature of the nucleophile and the nature of the alkyl halide. We don't know that yet, but we will. So let's talk about some effects of the structure of the alkyl halide on the rate of SN2 reactions. So I'm going to write some data on the board. I've got a reaction drawn out where an alkyl halide can react with iodide delivered in the form of lithium iodide. We're using acetone as a polar aprotic solvent in this reaction, and the iodide is just replacing the bromide. Okay, so organic chemists examined this reaction with 4 different alkyl halides: bromomethane, bromoethane, isopropyl bromide, and tert-butyl bromide. Okay? And they measured the relative rates of these reactions. And we're going to set the rate of the SN2 reaction with isopropyl bromide as 1. Okay, that's our standard rate. So this is a secondary alkyl halide. And it turns out that the rate of the SN2 reaction with a primary alkyl halide, bromoethane, is much faster, over 1,000 times faster than the reaction of the secondary alkyl halide. It also turns out that the reaction of the bromomethane is way faster, about 200 times faster than the rate of the primary alkyl halide, and over 200,000 times faster than the rate of the secondary alkyl halide. And the rate of the reaction with the tertiary alkyl halide is basically non-existent. Okay? For purposes of the class, we will say tertiary alkyl halides are unreactive in SN2 reactions. So what we have is a case where our methyl halides react faster than our primary alkyl halides, which in turn react faster than our secondary alkyl halides. And our tertiary alkyl halides are unreactive in SN2 reactions. Okay? So clearly, there's an impact of sterics, right? The more sterically hindered alkyl halides react slower, but we need to understand why this is the case. Okay? We can't just memorize this information. We have to understand why it is true. So the SN2 reaction only has one step, so it's only the rate of that one step that matters. And if we have differences in rates, that means we have differences in energies of activation. The reactions with the faster rates will have lower energies of activation. If we're comparing differences in energies of activation of different reactions, which species from each reaction should we be looking at? Reactants, products, something in between. What species should we look at to consider, to try to understand differences in energies of activation? Should we be looking at differences in starting materials, differences in products, or differences in transition states? So the differences, the activation energy is the difference in energy between what two species? Reactants in the transition state, okay? I'll tell you something, maybe it would have made it clear for you. The alkyl halides are all going to be of very comparable energies. So if there's a difference in activation energy, what is going to be different? The transition states would have to be different. So let's take a look at those transition states and see how they're different. So let's draw the transition state for the substitution of bromomethane, our fastest reaction. Okay, at the transition state, we will have partial bonds between the carbon and iodine, the carbon and the bromine, partial negative charges on those atoms, and then we will have 3 hydrogens arranged in this trigonal planar geometry. And we'll put brackets around it, and we'll put the double dagger from Henry Eyring there to show that this is a transition state. Okay? So let's compare this transition state to the transition state with isopropyl bromide. The reaction occurs, but it occurs 200,000 times slower than it does with bromomethane. Okay? So we're going to have the same partial bonds, the same partial charges, but the 3 groups that are attached to that carbon are different. Now we have 1 hydrogen and 2 methyl groups. Okay? So based on these reaction rates, we can conclude that this transition state would be substantially higher in energy than that one. Why? Okay, Claire? Okay, we would have increased steric strain in the transition state on the right relative to the one on the left. Why is that? Yeah, we have 2 methyl groups in the same plane. Here we have 3 hydrogens in the same plane. Those are small atoms. But now that we have 2 methyl groups and 1 hydrogen in the same plane, these methyl groups, they have hydrogens branching off of them. Those hydrogens can get close together in space. And so you're going to have increased steric strain in this transition state compared to this one. Okay? And so that higher energy transition state, due to more steric strain, is why the SN2 reaction of the more substituted alkyl halides is slower. Okay? Secondary slower than primary, primary slower than methyl. And if we were to try to draw the transition state for the tertiary alkyl halide, the tert-butyl bromide, we would now have 3 methyl groups in the same plane. And that transition state would be so high in energy that the activation energy would be prohibitively high. It would be too high of a barrier for us to climb over. And that's why SN2 reactions do not occur with tertiary alkyl halides. So here we see, I've drawn for you the transition states on the board. Here we see energy diagrams for these 2 reactions. The book used hydroxide instead of iodide as the nucleophile, but it's the same, the results are the same. We have a case where the energies of the starting materials are very, very similar. The energies of the products are very, very similar. But when we have the isopropyl bromide, the energy of the transition state is higher than when we use bromomethane due to that increased steric hindrance. That higher energy transition state gives us a higher activation energy, which gives us a slower reaction, a significantly slower reaction in this particular case. Any questions about that explanation? There's another way to look at this issue of steric hindrance. This slide here shows us some ball and stick models along with some electrostatic potential maps of our methyl, ethyl, isopropyl, and tert-butyl bromides. and it shows the nucleophile approaching from the backside. We know why the nucleophile has to approach from the backside. And we see that as we replace hydrogens with alkyl groups, as we make our alkyl halide more hindered, now not only would we have steric hindrance at the transition state, we have steric hindrance as the nucleophile is approaching. So before we get to the transition state, we have steric hindrance as well. With these alkyl groups pointing towards our nucleophile, it becomes more difficult for that nucleophile to approach the backside of the carbon at the proper orientation to cause the SN2 reaction to occur. Okay, so another way to look at it, we see that steric hindrance impeding the approach of the nucleophile, and that results in a slower reaction. Questions? So we have one of these nice summary tables that the book gives us, characteristics of the SN2 mechanism. As with all of these tables in our book, I urge you not to memorize the information in the table, but I urge you to understand the information in the table and to be able to explain why it is correct. So the kinetics, the second order kinetics, that was experimentally determined. As I told you on Friday, chemists ran experiments and they found that when we have SN2 reactions, the rate depends on the concentration of both reactants. Okay? So this was experimentally determined. The one-step mechanism explains the bimolecular kinetics. And then today we've explained why the SN2 reaction occurs with inversion of stereochemistry or backside attack. And we've also explained why less hindered alkyl halides react faster than more hindered alkyl halides. Okay? Any questions on those points? So SN2 reactions are very common both in the lab and in our bodies and in other living systems. So they occur in flasks and they occur in biochemistry. This slide here shows you a couple of SN2 reactions that are used to produce pharmaceuticals. Ethambutol is an anti-tuberculosis drug. And this example shows us that if we have a molecule with two leaving groups, it can undergo two SN2 reactions. So we would have two equivalents of this amine reacting with our dichloride, giving us this final product. Remember, I told you on Friday that ammonia, NH3, is always considered a strong nucleophile. And I told you that other amines, where you have NH2 bonded to an alkyl group, sometimes can function as a strong nucleophile. And this is one of those examples where this is functioning as a strong nucleophile, performing an SN2 reaction and giving us this product. And then down here we have fluoxetine, also known as Prozac, a very, very common medication. And there's a similar SN2 reaction with an amine, a methylamine here, primary alkyl halide. In both cases, they're primary alkyl halides, so these are going to be relatively fast SN2 reactions. And then one step converts the SN2 intermediate into the fluoxetine drug. Okay? So in the body, SN2 reactions are a little bit different. And that's because our alkyl halides are not water soluble. And so most of the reactions in living systems occur in aqueous solution. And so having alkyl halides that are not water soluble just doesn't work. And also it turns out that many alkyl halides are toxic. And so our bodies would not want to use them as the electrophiles in SN2 reactions. So instead of using iodomethane or bromomethane, when we want to attach a methyl group to a molecule via an SN2 reaction, our body uses something called S-adenosylmethionine, which is abbreviated as SAM. And SAM has our methyl group attached to a positively charged sulfur atom. And that's the part that's the leaving group. And now I'm going to draw the part that's not the leaving group, which is the majority of the structure. It's a very large molecule. Make sure I get this right here. Okay, we have, yep, that, and this, and the OH groups, and then we have a nitrogen, and then we have a nitrogen here, nitrogen there, NH2 here. Okay, so this is S-adenosylmethionine. Please don't memorize the structure. I'm not going to ask you to memorize the structure. As you know, in organic chemistry, we like to abbreviate things. So this is often abbreviated as R2S. We just use R groups to show these groups that are coming off of the sulfur. The purpose of all these groups is to provide water solubility for this molecule. We have lots of functional groups that can hydrogen bond to water, so this is most certainly going to be water soluble. Many of these groups can also engage in hydrogen bonding to functional groups at the active sites of enzymes that catalyze reactions, that catalyze these SN2 reactions. So that's also important. S-adenosylmethionine can fit into the active sites of these enzymes. This positively charged sulfur, when this molecule engages in an SN2 reaction, it's going to leave as a sulfide or a neutral sulfur, and that's going to be a very weak base. And so that will satisfy our requirement for having a weakly basic leaving group in an SN2 reaction. Okay? So this is nature's version of iodomethane or bromomethane or chloromethane. Question? The leaving group is everything that's not the methyl group. So what happens in an SN2 reaction with SAM is your nucleophile attacks the methyl group and it cleaves the carbon-sulfur bond, and all of this leaves. It seems very inefficient, but as I mentioned, there's a reason for that. It imparts water solubility, and it also allows these reactions. In biochemistry, we have to control when and where the reactions occur. And so if it can only occur at the active site of an enzyme where this thing is held in place by intermolecular forces to the enzyme, then we have control over the process. So there are reasons why nature uses something very complicated, that looks very complicated, as a leaving group. Okay? So this figure here is not very high resolution, but it's in your book, so you can take a look at it. And this shows you a reaction that occurs in your body anytime you get scared. So the fight or flight mechanism is initiated by formation of adrenaline. Okay? And adrenaline just has one more methyl group than noradrenaline, or its precursor. So what happens when you are scared or you have some other sort of stimulus that gets you in motion and action is that your nervous system sends a signal to your adrenal glands. Your adrenal glands are right by your kidneys to produce adrenaline. And so adrenaline is produced by SN2 reaction of noradrenaline with SAM. And then what adrenaline does once it's formed is it causes your heart rate to increase. It causes your blood pressure to increase. It causes the breakdown of amylose or starch and similar compounds to glucose, which can then be used as energy. And then it also causes your bronchial passages to dilate so that you can get more oxygen into your blood. So it causes all those things to happen, allowing you to generate energy fast enough to outrun the bear that's chasing you or whatever it is that's causing your adrenaline to kick in. Okay? So SN2 reaction occurring in biochemistry. Question? Yep. And it has a big effect. So we'll see this over and over again. When we look at biochemistry reactions, which we'll do frequently throughout the class, we'll try to blend organic chemistry and biochemistry. You'll see that very, very small differences in molecules can elicit very, very big differences in outcomes. For example, the hormones that determine male sex characteristics versus female sex characteristics They differ by the position of one methyl group and one hydrogen atom. Not much difference. So that's a common theme in organic chemistry, that small differences in structure can have big differences in outcomes. You just have to have them at the right places. So not all biological SN2 reactions are good. Here's a bad one that I'll show you. What does mustard gas do? What is it used for? Warfare. It's a chemical warfare agent. Fortunately, it's not used much anymore, yes? Okay, yeah, it can attack the nervous system. It can attack everything, basically. It's a very, very simple molecule. Mustard gas is this very simple molecule right here. The name is derived from the fact that it apparently smells like the mustard plant, although I've never smelled mustard gas, and I don't know anybody who has, so I don't have that on good authority, but that's what I've heard as far as where the name comes from. So an interesting thing about the structure of mustard gas is it has a nucleophile and two leaving groups in the same molecule. We'll learn more about the chemistry of sulfur-containing compounds in Chapter 9, but what you'll learn is that in some circumstances, a neutral sulfur atom can be a strong nucleophile. Okay? So when you have a nucleophile and a leaving group in the same molecule, you can have an intramolecular SN2 reaction to form a ring. And that's what happens here. We form this ring. This ring is strained, and it also has a positively charged sulfur. We just learned with Sam that positively charged sulfurs are good leaving groups. So this allows another nucleophile to come in an SN2 reaction that causes our good leaving group to depart and releases the ring strain from that three-membered intermediate. Okay? So this happens very readily. Our DNA has plenty of nucleophilic nitrogen atoms on it. So strands of DNA can function as nucleophiles in our bodies. And then because mustard gas has a second leaving group, the same two steps can occur. We can do our intramolecular SN2 reaction, forming that three-membered ring, followed by the intermolecular SN2 reaction using another strand of DNA. And what we end up with is a cross-link. We're either cross-linking two strands of DNA, or we're forming a cross-link on one strand of DNA. The end result is the same. We're preventing that DNA from replicating. Okay? If we're cross-linking our DNA, we cannot replicate the DNA, and so we're preventing cell division, and we're causing death. Okay? So it's a very simple, yet very effective way of killing cells and ultimately killing people if you get a big enough dose of mustard gas. So a big enough dose will kill you. Smaller doses will have effects that may be reversible. You can be blinded. Sometimes that blindness is temporary. Sometimes it's more permanent. Mustard gas was used most extensively in World War I. Are there any history buffs who know the name of the most famous mustard gas victim? No, we need to study our history a little bit more, I guess. Adolf Hitler. Adolf Hitler was a soldier in the German army in World War I. He was part of a mustard gas attack. He was temporarily blinded, but he recovered. He's the one person on earth that I think all of us wish would have died in the mustard gas attack because the course of history would have been different, but he didn't. And because he didn't die, World War II happened, and in World War II, chemical weapons were used rarely, if at all, but both the Allies and the Germans had mustard gas and other chemical weapons. And in 1943, after the US invaded Italy and the war was starting to turn against the Germans, we received some intelligence suggesting that the Germans were going to use mustard gas. Okay? So what we did, President Roosevelt ordered us to ship our mustard gas to Italy so that we'd have it ready to use. And if the Germans found out that we had it, hopefully that would be a deterrent. They wouldn't use their mustard gas because they'd know that we would retaliate. That was the idea. Okay? Unfortunately, it didn't work out. What happened was the Germans bombed the ship that was in a harbor in Italy that had the mustard gas, unleashing this massive cloud of mustard gas that killed about 1,000 people in the port. Okay, so it was a tragedy. But believe it or not, out of that tragedy came the world's first anti-cancer drugs. And that is because there was a chemist in the Army named Lieutenant Colonel Alexander. And he was dispatched to the scene to figure out what happened. This was a fairly secret mission, and all the people who knew the precise identity of the cargo were killed. They knew it was probably a chemical warfare agent, but we didn't know which one we had secretly shipped to Italy. So we sent this Army officer there to investigate, and because he was an expert in chemical warfare agents and he examined the victims, it was pretty easy for him to tell that they had been victims of a mustard gas attack. But he noticed something that nobody had noticed before. They did some autopsies of the victims and found that their white blood cells were depleted. Many of them just had their white blood cells totally wiped out. And he knew, and it was known at the time, that white blood cells divide more rapidly than most cells. So Lieutenant Colonel Alexander noticed that mustard gas was killing cells that divide rapidly faster. When we see this mode of action, that makes sense. But this chemistry was not known in 1943. There were people investigating it, but nobody knew exactly how mustard gas was killing people. So, Lieutenant Colonel Alexander also knew that tumor cells divide more rapidly and grow more rapidly than normal cells. So he wrote a report of this incident, and in that report, he suggested that less toxic versions of mustard gas might be effective at killing cancer, killing tumors. In the 1940s, there were no anti-cancer drugs. It seems crazy to us today because there's tons of them, but we had no therapies for cancer in the 1940s. So there were two chemists at Yale. Their names were Goodman and Gilman. And they were researching the mode of action of mustard gas. They contributed to figuring out how mustard gas is killing cells and killing people. They read this report, and they immediately shifted the focus of their research to synthesizing analogs of mustard gas that would hopefully be less toxic, but would be toxic enough that they would be able to kill tumor cells. And they came up with Mustargen, or mechlorethamine, which was the first chemotherapeutic agent, the first anti-cancer drug, introduced around 1950. You see it's very similar to mustard gas, so it works by the same mode of action. It just has a nitrogen with a methyl group as opposed to the sulfur, and that changes the toxicity enough. It's still toxic to regular cells, but if you get the right dose, you're going to kill mostly the tumor. You're also going to kill the hair, but then you're hopefully not going to kill the patient. And then a follow-up on Mustargen or mechlorethamine was cyclophosphamide or Cytoxan, which now has this six-membered ring with a phosphorus, oxygen, and nitrogen attached instead of the methyl group. And this drug is still in use today. You know, it's probably 65 to 70 years old, but I know somebody who had a very aggressive form of breast cancer a couple of years ago, and one of the drugs she was treated with was cyclophosphamide. So it is still saving the lives of cancer patients even today. So it's kind of ironic that something positive came out of mustard gas. But the reasons we're talking about it now, one is that the mustard gas mode of action teaches us that you can have intramolecular SN2 reactions when you have a nucleophile and a leaving group in the same molecule. And the other thing we learned from this story is that many scientific advances are made when somebody connects two ideas that have never been connected before. So Lieutenant Colonel Alexander made a very keen observation. But he observed that the fast-dividing cells were being killed preferentially, and then he connected that to the idea that mustard gas or its analogs could kill tumor cells. So making those connections that nobody has made before is a really good way to make a profound scientific discovery. Any questions? All right, so let's talk about SN1 reactions now. And we drew an SN1 reaction for you last week. where we took a tertiary alkyl halide, and as you'll see, we'll talk more about SN1 on Wednesday. Tertiary alkyl halides are the most common alkyl halides in SN1 reactions. And we know that in the 1 stands for unimolecular. Only one of our two reactants is participating in the rate determining step. So we are breaking the bond between the carbon and the leaving group. In this case, it's a bromide. And that's generating a positively charged intermediate, which we call, a carbocation. And then that carbocation is reacting with our nucleophile to generate the product. Okay? Question? We will talk about that in a second, but to answer that, how many groups are attached to that carbon? 3. So when we have 3 groups, what's the geometry? Trigonal planar. Yes, it's trigonal planar, and we'll explore that in just a couple of minutes to why that's significant. Okay? So our first step is the slow step. Why is the first step the slow step? Yeah, all we're doing is breaking a bond. So we know that this carbocation is going to be higher in energy than this, and we're going to have to go over an even higher barrier in order to get to that intermediate. We also know it experimentally because when chemists were studying the kinetics of this reaction, they found that the reaction rate was independent of the nucleophile. You change the concentration of the acetate ion, and that has no effect on the rate. So we know that the nucleophile is not participating in the rate determining step. So we end up with this energy diagram shown here on the screen. You see that each step, we have 2 transition states, so we have 2 steps in the reaction. 1 intermediate, the carbocation. The energy of activation of step 1 is significantly higher than that of step 2, which is what we would expect, since we know step 1 is our rate determining step. And so this is general for carbocations. Carbocations are always going to be slow to form, and then they're going to be quick to react. So we'll see that. Anytime we have a carbocation intermediate, slow to form but quick to react. And that's because bond breaking is involved in forming it, and then it reacts via bond forming processes. We see in this case, this is overall exothermic. They didn't draw the byproducts. They didn't even draw the acetate nucleophile except here. But if you look over here, you see bromide is our byproduct. Acetate is our nucleophile. Bromide is a weaker base than acetate. And so indeed, our leaving group is less basic than our nucleophile. So that tells us we're going to have an exothermic, a thermodynamically favored reaction. Okay? All right, so we started out class talking about the stereochemistry of the SN2 reaction. What about the stereochemistry of the SN1 reaction? It's actually different from the SN2 reaction. We can have chiral alkyl halides. I'm going to draw one for you. It's going to be a tertiary alkyl halide because I told you that tertiary alkyl halides are the most common alkyl halides in SN1 reactions. Okay? So this tertiary alkyl halide has 3 different alkyl groups, a methyl group, an ethyl group, and a cyclohexyl group, plus the bromide makes 4 different groups. So this is a stereocenter. If we take a single enantiomer of this alkyl halide, so it's optically pure. Another thing you'll learn about SN1 reactions is weak nucleophiles are very common in SN1. They don't have to be weak. Acetate is a strong nucleophile, but many of our SN1 reactions involve weak nucleophiles, and water is a fairly common weak nucleophile. So what happens in this SN1 reaction is we get the enantiomer where we've inverted the stereochemistry. Okay. So far it looks the same as SN2 chemistry, but we also get the enantiomer where we've retained the stereochemistry that has the same stereochemistry as our starting alkyl halide. Okay? And we end up with a one-to-one mixture of these products. Okay? We get a racemic mixture. So this reaction is referred to as a racemization. Okay? A racemization is the formation of a racemic mixture from an optically active or optically pure starting material. So if you have one enantiomer of your starting material and you convert it into a racemic mixture of your product, that is called racemization. And that's a dirty word. That's not something you want to happen in organic synthesis. Usually if you have a single enantiomer, you want to keep it as a single enantiomer. You don't want to end up with a racemic mixture. Okay? And this is general. The book shows you a couple of examples of other racemizations in SN1 reactions. Okay? Racemic mixture. This is another one of those molecules where they use deuterium to make their alkyl halide chiral. Here's one with methyl, ethyl, and propyl reacting with water. You notice that when you use water as the nucleophile, your product is an alcohol. And we'll show you how that happens in a moment when we draw the mechanism. But first of all, why are we having racemization in this case? Any ideas? Yes? Okay. Okay, very good. So we need to look at the structure of the carbocation intermediate. The structure of the carbocation intermediate holds the key to understanding why we get racemization. So as we mentioned, a carbocation is going to be planar, trigonal planar, and it's going to have an empty p orbital. So its three groups are going to be arranged in this trigonal planar orientation. And then the new bond is going to form when a pair of electrons on our nucleophile will come in and overlap with this empty p orbital. So that pair of electrons could come either from the bottom or the top with equal rates. It's not going to see a difference between either face of this achiral carbocation. So since that nucleophilic attack in step 2 occurs with equal rates, we get a one-to-one mixture of the enantiomers. So we'll go ahead and draw that on the board for our example alkyl halide, our cyclohexyl, and then we'll also show you how we end up getting the alcohol. Okay, so we're going to lose bromine. I'll just go ahead and draw the bromide right here. And then I'm going to draw this carbocation on its side. That wasn't a very good p orbital. Okay, so here's our carbocation that we get. Then we have our water molecule. And that water molecule can attack the carbocation either from the left or from the right. Is there going to be any difference in the rates? Is that water molecule going to see anything different when it comes from the left or the right that would cause one mode of approach to be faster than the other? It will not. So I'll go ahead and draw dashed arrows going in both directions, indicating that sometimes it will come from the left, sometimes it will come from the right. So when it comes from the left, we're going to end up with a positively charged oxygen. That's known as an oxonium. And then we have our ethyl group and our methyl group, okay? And then whenever we run reactions in water, we're going to have an excess of the water. Whenever we use it as a nucleophile, we have excess water that is present. So the excess water molecules that didn't participate in the substitution will then, one of them will deprotonate this oxonium ion, and that's how we get our final product that just has the OH group. Okay, and we generate H3O plus. That H3O plus just forms a salt with our bromide ion. Okay, and this step is very rapid. Proton transfers between two heteroatoms, two oxygens in this case, are some of the fastest reactions we have in organic chemistry. So this occurs very rapidly, giving us this alcohol product. Question? You'd have very similar pKa's of this versus that. So it's a concentration issue. This molecule is swimming in a sea of water molecules. And so that's going to cause that proton transfer to be highly favored. Very good question. Okay, so we do the same thing from the right side. And what we're going to end up with is the mirror image. And then our water molecule functioning as our Bronsted-Lowry base in this final step, deprotonating the product, or deprotonating the intermediate to afford the product. We get our H3O plus. And so because the rate of the water attacking this carbocation is the same from the left versus the right, we get a one-to-one mixture of these intermediates, which then get deprotonated to give us a one-to-one mixture of our product enantiomers. So the racemization that occurs with in an SN1 reaction is due to the generation of an achiral carbocation intermediate. So last thing I'll show you for today. Whenever you have an achiral intermediate, you can produce a chiral product, but it's going to be a racemic chiral product, okay? The alcohol that we are getting in this reaction is chiral. It has a stereocenter, but it's being produced as a racemic mixture. So this is also general. If we have an achiral intermediate, we're going to form a racemic mixture. All right? Any questions? All right, we'll pick up there and talk more about SN1 reactions on Wednesday.